Hello everyone, I'm Yuki Okawa from Osaka International Cancer Institute in Japan. I am a researcher belonging to Dr. Naoyuki Taniguchi Laboratory and I am listed as second author in this poster presentation. This time I would like to talk about keratin sulfate disaccharide about specific targeting to rangerin and possible application to COPD. Keratin sulfate is one of the proteoglycans expressing mainly in joints to be the cushion by tissue hydration. There are three types of keratin sulfate, Ks1, Ks2, and Ks3, and all types of keratin sulfate are composed of racemic disaccharide, a galactose beta 1,4 linked to the N-acetylglucosamine. As you can see, this disaccharide is often sulfated. We call this sulfated disaccharide as L4. On the other hand, COPD, chronic obstructive pulmonary disease, is the third commonest cause of death in the world in 2019. As you may know, the majority cause of COPD is tobacco smoking. Tobacco smoking damages the airway and induces inflammation. Chronic inflammation leads to severe obstruction of the airway, which impairs functions of lung for gas exchange. Keratin sulfate is also expressed in the lung, and importantly, this immunohistochemical analysis revealed that the expression levels of keratin sulfate is reduced both in the human lung of COPD patients and in the mouse lung after 3 months smoking. These results suggested the hypothesis keratin sulfate has protective functions against the lung, which prevents COPD onset and its development. Following this hypothesis, we focused on keratin sulfate disaccharide L4. In the previous study, we identified rangerin as a binding partner of L4. Rangerin is a C-type lectin receptor and expressed in the Langerhans cells in the skin and dendritic cells in the lung. This is the primary structure of mouse rangerin. This is type 2 membrane protein on the plasma membrane. Actually, keratin sulfate disaccharide have different sulfation patterns. Notably, we found L4 and L3 can bind to the rangerin in vitro assay. Following this, and to produce the more effective compound, we prepared L4 derivatives, for example, triangle and polymer form by chemical synthetic method. Interestingly, in the competitive ELISA assay, L4 derivatives showed the binding affinity much stronger than L4 monomer. The IC50 value competitive to L3 are 2.7 nanomolar for triangle L4 and 2.1 nanomolar for L1 polymer. We think this nanomolar scale of IC50 of L4 polymer is preferable to develop a drug candidate for clinical care. And next, we tested anti-inflammatory effect of L4 and the derivatives in rangerin expressing BMDC, mouse bone marrow derived dendritic cells. After incubation with them, we examined the gene expression levels of inflammation-related cytokines by RT-qPCR analysis. As you can see, we found that downregulation of inflammatory cytokines IL-6 and TNF-alpha, whereas upregulation of anti-inflammatory cytokine IL-10 by incubation with L4 and the derivatives. Following this, we next challenged L4 treatment in COPD model mice. In this mouse model, the administration of PPE, porcine pancreatic elastase by intratracheal spray can induce COPD-like phenotypes within a few weeks. PPE administration following L4 administration was performed and low attenuation area of the lung indicating emphysema area were measured by micro-CT analysis. As you can see, L4 administration reduced low attenuation areas significantly, which indicates L4 mitigates COPD phenotypes in vivo. We also performed histological analysis to confirm the protective functions of L4 administration in mice. HE staining of the lung demonstrated that L4 reduced linear intercept of pulmonary alveoli. This is a summary of this study. We produced keratin sulfate disaccharide L4 and derivatives such as triangle and L4 polymer. These oligomeric L4 derivatives bind to rangerin stronger than L4 monomer and downregulate the expression of inflammatory cytokines. This functions L4 mitigates COPD phenotypes in mice. Finally, we show you future plans. To develop novel L4-based therapy for COPD, more detailed molecular functions of rangerin should be demonstrated. For this, we will study to clarify the downstream signaling of rangerin and evaluate the effect in human COPD patients. Thank you for watching our presentation. Please feel free to contact us if you have some questions about this study. Thank you very much.